Immunology Insights, Basics and Beyond, Chapter 20, Antigen Presentation. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we explored phagocytosis, a crucial mechanism that enables immune cells to engulf and digest pathogens. But what happens after these pathogens are broken down? How does the immune system recognize and respond to specific threats? That's where antigen presentation comes in. Consequently, today, although touched upon before, we will discuss the process of antigen presentation in greater detail, the molecular players involved, and how it bridges innate and adaptive immunity. Understanding this mechanism is fundamental to comprehending how vaccines work, how T cells get activated, and how our immune system distinguishes between self and non-self. So let's talk science. What is antigen presentation? Well, antigen presentation is the process by which immune cells display fragments of pathogens, called antigens, on their surface for recognition by T cells. This is a key step in activating the adaptive immune system, allowing it to mount a precise and effective immune response against invaders. There are two main pathways of antigen presentation. First, the MHC class I pathway or endogenous pathway. This pathway processes intracellular pathogens such as viruses or tumor antigens. Inside the cell, intracellular proteins, including viral and tumor proteins, are degraded by the proteasome into small peptides, which are then transported into the endoplasmic reticulum or ER by TAP, a transporter associated with antigen processing. Here, peptides are loaded onto major histocompatibility complex or MHC class I molecules and transported to the cell surface. These antigens are recognized by CD8 positive cytotoxic T cells, which then eliminate infected or tumor cells. Second, we have the MHC class II pathway or the exogenous pathway. This pathway processes extracellular pathogens such as bacteria and fungi. These pathogens are engulfed via phagocytosis or endocytosis, then broken down in lysosomal compartments. The resulting peptides are loaded onto MHC class II molecules in specialized vesicles and presented on the cell surface. These antigens are then recognized by CD4 positive helper T cells, which then orchestrate a broader immune response by activating other immune cells, including B cells and macrophages. What are the key players in antigen presentation? Well, several immune cells specialize in antigen presentation, known as antigen-presenting cells or APCs. First, we have dendritic cells. They are the most efficient APCs, crucial for initiating adaptive immune responses by activating naive T cells. They act as messengers between the innate and adaptive immune systems. Second, we have macrophages. They are primarily involved in antigen presentation to memory T cells, reinforcing immune responses during secondary infections. And third, we have B cells. They produce our antibodies. As APCs, they capture and present specific antigens to helper T cells, facilitating their own activation and promoting their own antibody production. This is, as in the other described cases in previous chapters, a fail-safe mechanism to avoid unregulated antibody production. The helper T cells confirm the antigen and help activating the B cells and their antibody production. Each of these cells presents antigens to shape an effective immune response, ensuring that only relevant T cells are activated. Now, let's have a closer look how antigen presentation works. First, we have the antigen uptake. APCs capture pathogens or their fragments via phagocytosis, endocytosis, or receptor-mediated uptake. Second, we have antigen processing. Intracellular proteolytic enzymes break down the pathogen into smaller peptide fragments within compartments such as the proteasome for the MHC class I pathway or the lysosome for the MHC class II pathway. Third, we have the MHC loading. Peptides are loaded onto MHC molecules within distinct intracellular compartments. MHC class I molecules bind peptides in the endoplasmatic reticulum or ER before being transported to the cell's surface. MHC class II molecules bind peptides in specialized endosomal vesicles before transport to the membrane. Fourth, we have the antigen display. The MHC antigen complex is transported to the cell's surface, where it is recognized by the T cell receptors or TCRs on the T cell surface. Fifth, we have the T cell activation. The interaction between an APC and a T cell combined with co-stimulatory signals, for example CD80, CD86 binding to CD28 on T cells, determines whether the T cells becomes activated and initiates an adaptive immune response. Antigen presentation plays a role in both health and disease. Antigen presentation is essential for effective immune defense, but errors within the process can lead to severe consequences. First, we have autoimmune diseases. Improper antigen presentation can cause the immune system to target self-antigens, leading to conditions like multiple sclerosis or MS, where myelin is attacked, and rheumatoid arthritis or RA, where joints are inflamed due to immune attacks. Second, we have cancer immunoevasion. Some tumors escape immune detection by downregulating MHC class I molecules, making them invisible to cytotoxic T cells. Immunotherapies such as immune checkpoint inhibitors, for example PD-1, PD-L1 therapies, work by restoring T cell activity against tumors. Third, we have viral evasion strategies. Viruses like HIV or the cytomegalovirus or CMV interfere with antigen presentation by blocking the TAP transporters or degrading MHC molecules, allowing them to persist undetected in the body. Of course, there's also therapeutic exploitation of these mechanisms. Many vaccines work by enhancing antigen presentation, ensuring a strong T cell response. Cancer vaccines aim to improve tumor antigen presentation to boost anti-cancer immunity. CAR T cell therapy is another approach where T cells are engineered to recognize specific tumor antigens independently of MHC presentation. To conclude, antigen presentation is a fundamental link between innate and adaptive immunity, ensuring that our immune system effectively recognizes and fights infection. It enables T cell activation, immune memory formation, and precise immune targeting of threats. Without this process, pathogens would evade detection, immune responses would be ineffective, and the body's ability to fight infections, tumors, and autoimmune conditions would be compromised. For example, dendritic cells play a crucial role in initiating immunity. Macrophages reinforce ongoing responses, and B cells optimize antibody production. MHC class I and class II pathways ensure that both intracellular and extracellular pathogens are accounted for, providing comprehensive protection. In the next chapter, we will explore the clonal selection and expansion, the mechanism by which activated T cells multiply and differentiate to eliminate threats efficiently. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So see you next time and thank you for listening.